Hi, everybody. People are trickling in. It usually takes a few minutes for everyone to join us. Welcome, welcome. See, people are still coming in the room. It takes a few minutes usually for everybody to arrive. Maybe we'll give folks one more minute to come in on this sort of cold, unseasonably cold, late May evening. But grateful to spend time cozily indoors with all of you, learning about science. Bob, I was looking at your slides and I'm very excited about them. Right. Well, I think more people will probably trickle in, but it's probably safe to kick things off. Hi, everyone. Good evening. My name is Rachel Rosen Simpson. I'm the program manager at the Breast Cancer Coalition. I'm so glad that you could all join us this evening to learn more about understanding the latest advances in cancer testing with Dr. Bob Weiss. We're grateful to the Lisi Rosenthal Memorial Education Fund that supports coalition educational offerings just like this one, because we couldn't do it without them. Before we begin this evening's presentation, there's just a few housekeeping announcements to share. Anyone who would like closed captioning, it's available if you click the CC button at the bottom of your Zoom screen that says Show Captions. If you missed the link in the Zoom reminder email for accessing a PDF of Dr. Weiss's slides so you can follow along with the presentation, I'll drop that link into the chat for anyone who would like it. Maybe I'll do that quick right now. So in the chat, here we go. To everyone. There's that for all of you. I will facilitate a question and answer session following the presentation. So as you have questions, please type them into the chat and I'll continue to compile them as the presentation unfolds. I'll share them at the end with Dr. Weiss and we will answer as many as possible before we try our best to wrap up by 8:30. We always try to get everybody out of here by 8:30, maybe sooner. We'll see how many questions you have. A disclaimer that Dr. Weiss cannot give any specific medical advice for an individual case during this short time we have together this evening. So if you could try to keep your questions broad and inclusive of others, that would be very much appreciated. This presentation is being recorded, so if you want to watch it again, it's being added to our webinar library probably by the end of this week, as well as uploaded to our YouTube channel, and it's publicly searchable now, so you can share widely. I'll drop the links to our webinar library and the link to register for our future webinars into the chat. Please join us in the future. And then there will be a survey that pops up on your browser at any point when you leave the webinar. And we really do utilize your anonymous feedback when planning our program offerings for the future. So please, please complete the survey. It's just 10 questions. It's really quick, and that will help guide us in the future. So anyway, tonight's webinar features a very special guest and a longtime friend of the coalition, Dr. Bob Weiss. Welcome, Bob Weiss. Many of you may know Dr. Weiss through his unwavering commitment to our mission at the coalition. He has served as a valued member of both our advisory council and our research advisory board, and he's played a key role as a scientific expert during our annual research review day. Dr. Weiss is a respected researcher and educator at Cornell University, where his lab is at the forefront of scientific discovery. The Weiss Laboratory develops mouse models to study how genomic instability and metabolic dysfunction affect mammalian development and tumorigenesis. Through his work, Dr. Weiss and his team are helping to uncover critical insights into the biological mechanisms that drive cancer. And in addition to his groundbreaking research, Dr. Weiss is instrumental in mentoring future researchers. His dedication to education and mentorship helps ensure the next generation of scientists is equipped to continue advancing the field of cancer research. Tonight, Dr. Weiss will guide us through some of the latest advances in diagnostic and treatment monitoring tools used in breast and gynecologic cancer care. And it's a fantastic opportunity to deepen your understanding of many cutting-edge developments and hear directly from a reader in cancer research. And now, without further ado, take it away, Dr. Weiss.
Okay, thank you, Rachel, for that really nice introduction. And hi, Christina, and hi, everybody that's online tonight. I can't see all the attendees, although I glanced quickly at the participant list and see a lot of familiar names from serving on the grant review panel. I think super highly of the Breast Cancer Coalition of Rochester, how you support research, how you have lots of outreach and educational programming and support cancer patients all throughout New York. So I really appreciate all that you do. I'm going to go ahead and share my slides. So we have a pretty large topic today focused on latest advances in cancer testing. And there's a lot happening in this area, and I'll cover several aspects of this. There's a lot happening in many different avenues, and I won't have time to talk about everything, but I'll do my best to introduce you to some key concepts and new advances that I think are already impacting clinical care. And I thought I'd actually start with a picture of my laboratory at Cornell University. And I show this to remind myself to mention a couple of things. As Rachel just mentioned, I have a research lab that focuses on fundamental mechanisms in cancer biology. So I'm a molecular biologist and a mouse geneticist. And as you heard, I'm not a clinician. So I've never diagnosed a patient. I've never prescribed a test like the ones I'm about to talk about. So if you have clinical questions, please consult your doctor. And I'll focus on the mechanism and the underlying science and try to explain how some of these advances are moving forward. The other thing I wanted to note, just related to kind of our current times, you can see this slide also lists some of the funding that I've been fortunate to have over the years from the NIH, National Cancer Institute, and National Institute for Child Health and Human Development. And I just wanted to mention, I think probably most people are familiar with this, but this is a really challenging time for the research community, for research and academia in particular. I've experienced this firsthand. So my graduate student, the first one listed there, Gerardo, had his predoctoral fellowship terminated prematurely. We at Cornell, when we get an NIH grant, we do the work, we submit invoices to the NIH for payment of our research activities. And these are research activities, these are not gifts from the federal government. These are things that we've competed for through a really rigorous scientific review process after the government has called for proposals. When we receive one of these awards, we then do this research under a contract and we submit invoices to get paid for reimbursing the cost. So Cornell has not been reimbursed for any of its NIH projects for the last 2 months, and it's not clear when that's going to end. So, and when support for our contracts is going to resume. So it is a challenging time. And what I'm about to tell you on the research front, essentially everything I'm going to describe in one way or another was made possible through research support from the federal government. The federal government supports research in such a critical way historically, both in driving the studies and then also training the future scientists. And I think both of these things are in jeopardy right now, the way the government is approaching research funding in academia and more broadly. So I just wanted, I'm sure people are aware of this, I just wanted to share my own experience with that. I think this audience that's joining us tonight is one that's especially interested in cancer biology, understanding mechanisms behind cancer, understanding how we can better treat cancers and how we can improve outcomes for people with cancer. All of that's going to require ongoing federal support. if you think that's the case, I encourage you to talk to your congresspeople, get out and vote, and hopefully we can keep our scientific enterprise, which is really a crown jewel of the United States. The biomedical research enterprise in academia in the United States is one of the greatest things in our country, and I hope we continue to support it. So with that, I'll go forward with my presentation. I did want to start with a little bit of background information on cancer and cancer mechanisms, kind of a cancer biology 101, just so we're all on the same page, just spend a quick few minutes on some of the underlying mechanisms. And as a molecular biologist, I'll start with kind of the fundamentals of how our cells work, because this is really critical to how we do cancer testing. And what I'm going to show here is referred to as the central dogma of molecular biology. So each of our cells contains DNA. This is the, the blueprint, the instruction manual for how cells operate. And this DNA, through the process of transcription, leads to RNA molecules. And then RNAs are translated to make proteins, and proteins are kind of the, the engines of the cell, the doers, the machinery are the proteins. So the DNA is the instruction manual. RNAs are kind of the intermediate molecules, and the proteins, for the most part, are doers. This is a vast oversimplification, but that's generally how things work. And I'll refer to some molecular approaches that we use then to characterize cells at each of these levels. So genomics refers to the study of all genes within a cell. So now we have the technologies, we can essentially simultaneously study each of the 25,000 genes that's present in each of our cells. We can also do more focused molecular assays to look at one or two genes at a time. And some of the diagnostics that we'll talk about are at the whole genome level. Others are more focused on specific genes. We can do similar studies at the RNA level. So we can measure, for instance, how much of a given gene is expressed in a cancer cell versus a normal cell. And sometimes those changes are very indicative about the behavior and the biology of a cancer cell. And we can use techniques like transcriptomics to study the level of every RNA in a cancer cell. And then we can use proteomics similarly to measure essentially every protein within a cell. So this is kind of the fundamental paradigm and how we can use it scientifically to measure what's going on in cells. This is a very simple version of the central dogma. Over the years, we've evolved improved understanding of all the complexity. So, this central dogma from the 1950s of DNA to RNA to protein has become more and more complex, and that kind of accounts for all the complexity we see, the variation between cell types, for instance. They all have the same DNA instruction manual, but a neuron in our brain, a muscle cell, a skin cell, right? Those all behave very differently, even though they have the same instruction manual. And that's because of differences in how they express genes, but then all of these other more complex processes that also vary from cell to cell. And these are kind of processes that often get deranged in cancer cells as well. And I just mention this because some of these more detailed molecular mechanisms like microRNAs listed here end up being really useful as diagnostic markers in cancer as well. Okay, so another fundamental issue related to cancer biology, so again, we're all on the same page, is that cancer is a genetic disease. And this slide kind of lists some of the key features to keep in mind. So somatic mutations occur in most cancers. So somatic mutations are mutations that occur in cells essentially after birth as kind of a random process where the DNA sequence in a cell changes. And that change sometimes can give that cell a growth advantage where it divides more aggressively. And that mutation that's arisen spontaneously in that cell then can drive the development of a cancer. So that's a common type of mutation, a somatic mutation that's arises spontaneously in a cancer. The other type of mutations that are found in cancer are what are called inherited germline mutations. These are mutations that an individual has inherited from either their mother or father. So those mutations are in every cell of that individual. And some of those mutations or variations actually are associated with elevated cancer risk. And there's some rare familial cancer syndromes where these inherited mutations lead to a very high risk of cancer. And we'll talk more about how the genes and the variation that we inherit from our parents can influence the risk of cancer. So it's really this accumulation of mutations, a combination of inherited variation and then these somatic mutations that can arise in cells. The accumulation of those mutations gives rise to cancer. And one kind of thing that follows with that is if there's ever an increase in mutation rate or genomic instability, this is something my lab studies frequently, those increased frequency of mutations then lead to increased frequency of cancer. Aneuploidy, in addition to mutation, is another hallmark of cancer. So in addition to mutations, little changes in the sequence of the DNA, one can also observe in cancers changes in numbers of chromosomes. So chromosomes can be gained or lost in cancers. That also is a common event in cancers. And then finally, one really important thing to consider is that cancer really is a disease where genetic selection is happening at the level of single cells. So when cells acquire a mutation, if that mutation gives the cell a growth advantage, that cell is going to grow and essentially outgrow its neighboring cells because of that growth advantage. So cancer really is a case of natural selection at the cell and molecular level. And we'll talk about this in the context of some of the resistance mutations. If you have a tumor that's getting a treatment, and a cancer cell within that tumor develops a mutation spontaneously that causes resistance to that treatment, that cell is going to have a growth advantage because it's going to be able to continue growing despite the presence of the treatment. And that's a kind of classic example of how this kind of Darwinian selection is happening in cancers all the time. And we'll see how that influences the origins of some of the resistance mutations that it's important we're able to detect and screen for as patients are receiving treatment. Okay, so cancer is a disease then of mutation, as I mentioned. Some of these mutations will affect genes that promote the growth of cells. They're called oncogenes. They're actively driving the cell to proliferate. And then some of the mutations happen in genes that are called tumor suppressors that are normally kind of restraining the growth. So you can either get activation of oncogenes and essentially the gas pedal being pushed forward. And then at the same time, you can get also mutations of tumor suppressors, the brakes that would normally restrain growth. And that combination of pushing on the gas, losing the brakes, causes the cancer cell to drive forward. And the number of different kinds of mutations that can happen in cancer is kind of astonishing. This table or chart kind of lists each row here is a different gene, and then across the columns are different types of cancer. I think breast cancer here is the second column, and you can see that some gene mutations are very common in many types of cancer. So for instance, here we have the p53 tumor suppressor gene. It's one of the most frequently mutated genes in cancer. Other mutations are selective. You'll see boxes that light up for particular cancers, CDKN2A, for instance, or other many APC is, for instance, a gene that's commonly mutated specifically in colon cancer. So I kind of wanted to show this just to illustrate that the complexity of cancer, that cancer is not one disease. It's really thousands of diseases, depending on the tissue type of origin or the cell of origin for the cancer, and then the specific types of mutations that are found in any one cancer. And the diagnostics that we're going to talk about are really kind of looking forward to the current era, which has gone beyond just treating cancers based on a cell of origin and really treating cancers in a more personalized individual manner based on the specific mutations that they have. And those mutations will influence how well a given cancer will respond to a given therapy. So it's really important that we understand for a given cancer, what are the driving mutations? What are the particular changes in that tumor cell? In many cases, not always, those mutations aren't always actionable, as we say, but some of them are, and that can drive how we treat the cancer. And so a lot of the technologies I'll talk about are how can we identify the very specific mutation in an individual tumor to guide therapy. There's also some really interesting data that's emerged from genome sequencing. So I mentioned that genomics, the study of all the genes, all the DNA sequence in a cell. And this now has been done for many cancer samples. And what we see is that there's specific patterns of mutations that emerge. There are those mutations in the genes that drive cancer, as I showed on the previous slide. There are additional mutations throughout the genome that are happening that interestingly can inform us about how the cancer arises. And I'm not going to go into the details here. These are just on the top rows here. You can see the specific types of changes in the 4 bases of DNA, right? We have A, C, T, and G. Those are the 4 bases of DNA. And in a genome, we have 3 billion pairs of those bases. And you'll see that in cancers, you'll see very specific patterns of certain kinds of changes in the DNA sequence. And what we've realized is that those specific patterns of changes actually reflect how that cancer arose. And so you'll see signatures associated, for instance, with smoking and tobacco mutagens, other signatures associated with exposure to sunlight and ultraviolet light, others related to alkylating agents. This is a little secondary to what I'm talking about, but I think it's a fascinating area of the biology, how we can look at the DNA sequence of a cancer, learn, as we'll talk about today, about how to treat it effectively, but also learn about how that cancer arose. And then hopefully we can use that knowledge in the future to maybe prevent exposures, limit the origins of cancer for other people in the future. Okay, so in the first part of the talk, I'm going to talk a little bit about some technologies that allow us to identify individuals that are at high risk of cancer. And I thought I'd start with a fundamental piece of data that I think probably this concept is something that everybody's familiar with, which is that early detection of cancer favors better outcomes. So when cancers are detected early at an early stage, they're typically more effectively treated. Later stage, more advanced cancers are more resistant to treatment and are associated with poor outcomes. So here you can see breast cancer stage at diagnosis. Early stage cancers have a very high level of 5-year net survival. And as the stage at diagnosis worsens, the outcomes worsen. So this indicates that we need technologies that allow us to, 1, identify individuals that are at risk of cancer, and then 2, detect cancers in those individuals. So I'll start with a little bit of information about what determines risk for individuals for developing a cancer at some point. And that risk is really a combination of genetic factors and then environmental factors and behavioral factors. And I'll focus here really on the genetic factors. So again, the DNA of any given individual is inherited from the parents, of course. So half of our chromosomal DNA comes from mom, half comes from dad. Each of our cells reflects that DNA sequence that we inherited from our parents. And among that DNA are variations in different genes. And each of our cells then carry those variations in particular genes across the genome. And some of those genes variations, they make all of our traits, right? So the variation in one gene might influence my hair color or my height or other features of my personality, et cetera. So those variations across our genome make the differences between individuals. They can also influence cancer risk. Some of those variations might impact the function of those genes that I described earlier that regulate how cells divide, whether it's a gene that functions as kind of that accelerator pedal or the brake pedal. If the function of that gene is maybe slightly altered by one of these variations, that can influence the risk of cancer in an individual. And so this slide kind of depicts how that variation impacts individuals, both in terms of the frequency of those variations in a population and then their effect size. How strong an effect are they going to have on cancer risk? So what you see in this graph, there are some variations that are very rare but can have a high effect size. So these are rare alleles causing Mendelian disease. So these are variations that an individual can inherit. They're not very common in the population. They're very rare because they're going to have some negative impacts. And these kind of rare high effect size genes could predispose an individual to cancer. So a BRCA1 or BRCA2 gene mutation would fall into this category. A relatively rare occurrence, but it can have a high impact on predisposing to disease, cancer in this case. There are some kind of intermediate effect genes. So these happen at lower frequency and they have lower impact on the likelihood of a phenotype like cancer risk, but they can still have a substantial effect. And then finally, there are very common variants that happen in many people and associated with very low risk of disease. And so if we look at this in the context of breast cancer, kind of same kind of plot, the frequency of these alleles along the x-axis here, and then the risk estimate or the effect size. How strong is their effect on cancer risk on the y-axis? And again, you see very rare genes like mutations or variations in BRCA1, BRCA2, P53. These are uncommon individuals, but if an individual does inherit one of these, that can have a very high effect size, a strong predisposition to cancer in that individual. So rare, high risk. Then there are these intermediate risk alleles, and you might recognize some of these gene names as being associated with breast cancer. And then finally, there are these rarer variants that are more common and have less of a strong effect on cancer, but still can be associated with cancer predisposition. And across the genome of an individual, one could inherit variation in multiple genes. And really the genetic predisposition, the genetic risk for cancer then would be that combination of inherited variation across the entire genome, combined with environmental exposures that might further increase risk. That could be, exposure to sunlight for skin cancer. It could be alcohol consumption for liver cancer. There can be other factors related to obesity or lack of exercise that also can influence cancer risk. So it's a combination of all those features that's going to drive risk. And I thought I'd point out this is one of many examples of what we call a risk calculator. So there are now tools that can combine for an individual, if there's information available through genome screening about what's the array of genetic variation that individual has, that information can be combined with consideration of other risk factors. And this is one particular risk calculator from this one that's developed at the University of Cambridge. There are many of these out there, but you can see that many factors, risk factors are calculated into this model. Family history of breast cancer, age at diagnosis of affected family members, the genetic variation, these high penetrance or high strong effect genes like BRCA1, BRCA2, but also less common genetic contributors that cumulatively could influence risk. And then some other factors like BMI, alcohol use, et cetera. And so all of these genetic and non-genetic factors can then be used to calculate a lifetime risk score. And one can imagine how this is useful then for deciding how to proceed with various clinical parameters. And this is for breast cancer, risk then we can think of in this kind of bell-shaped curve with, majority of individuals falling in this middle intermediate risk category. And then we have some individuals at this end of the curve that have a low risk of developing breast cancer. And then other individuals at this end of the curve, because of their genetic and other factors, have a very high risk of developing breast cancer. So this is all considering things before the cancer actually arises, but this can influence how we monitor for the presence of cancer. So for instance, an individual with a very low lifetime risk based on these parameters maybe would do mammogram screening less frequently and starting at an older age and have less frequent breast exams. Whereas somebody in the intermediate range would start screening earlier, do it more frequently, and under some conditions, consider other imaging tests. And then individuals that are at a high risk, then they should start screening early, use more sensitive imaging technologies like MRI, or maybe some of the molecular diagnostics that we're going to talk about today, and think about preventative actions, whether that's chemoprevention or surgical intervention. So I think this is a useful way of thinking about before cancers arise, how can we screen for risk and tailor approaches appropriately. So the next set of tests that I'll talk about are ones that apply once a cancer has been diagnosed. And I'm going to focus primarily on molecular analyses, I think exclusively on molecular analyses. I'm not going to talk about imaging as a really critical diagnostic tool for breast and other cancers. I'll focus on the molecular techniques. What we see on this table is several examples of different molecular techniques for early stage breast cancer. And some of these have very similar approaches. I'm just going to I picked a couple of these to talk about in a little more detail as examples. And I didn't do any disclosures at the beginning. I don't invest in any of these companies. I don't have any stock or other connections to any of these companies as an advisor. So I'm just giving a neutral presentation here. And again, I'm not recommending any of these tests for utilization. I think if you're interested in some of these tests, which in some cases are looking at different aspects of disease and depend on the specific type of disease, I'd really encourage individuals to talk to their clinician about the opportunities. But I'm going to talk a little bit about Oncotype DX and then also about this BCI or breast cancer index as a couple of tests that can be done on earlier stage breast cancers. And we'll start with Oncotype DX. So this is a test that's used for newly diagnosed early stage invasive breast cancer. So this can be done with node negative or 1 to 3 positive nodes. This is really intended for ER positive, HER2 negative cancers. So advanced metastatic disease, HER2 positive disease, triple negative disease, this is not the test to use. It's this specific scenario. And the purpose of this test is to predict the likelihood of chemotherapy benefit and the 10-year risk of distant recurrence. So again, these are ER positive patients, and the question here is really who's going to benefit from chemotherapy to prevent later stage recurrence. And the assay is based on a 21 gene recurrence score assay. And the way the technique works is, and I'll explain what this means in a second, RT-PCR assay with RNA isolated from fixed paraffin-embedded tissue. So when a tissue sample was taken from a patient's tumor for tumor histopathology, that tissue is fixed. It's embedded in paraffin wax, it's sectioned, and the pathologist reads that section for initial diagnosis. Quite remarkably, that very small amount of tissue in one of these sections can also be recovered. It's possible to isolate RNA molecules from that and to conduct an assay to determine the expression level of genes of interest within that sample. Excuse me. And this particular assay uses this 21 gene panel. The 21 genes that are measured, so the level of these genes in this tumor specimen is measured, and these include markers of cell proliferation, invasion, HER2 status, estrogen and hormone signaling. Then there's a set of what are called reference genes. Those are kind of for calibration of the assay. And then there's a few other genes that are included as well. And so based on the levels of the expression of these genes that is measured in these individual samples, there's an algorithm that was developed by the scientists that created this assay that predicts the likelihood of recurrence. And here's a little bit of data from one of the very early studies published in Lancet Oncology using this test. And this is from a clinical trial, SWOG 8814. There's been additional clinical testing since then. And you can see that in this particular clinical test, there was a comparison group. And that comparison group was patients treated with tamoxifen alone. So again, these are ER positive patients treated with tamoxifen alone versus chemotherapy. So cyclophosphamide, doxorubicin, and fluorouracil, this CAF combination therapy before tamoxifen. So in each of these graphs, we're going to compare two groups, the group that received tamoxifen alone versus that CAF tamoxifen group. And you can see that these data are stratified across the score that came out of the Oncotype DX test for each of the patients involved. And so in this upper left graph, these are ones that had a low recurrence score from that 21 gene signature. So the algorithm said there's a low risk of recurrence here. And you can see that for these patients with the low risk score, there was really very little, no significant difference between for outcomes at 10 years between patients that received tamoxifen alone versus the chemotherapy before tamoxifen. So these patients with the low recurrence score didn't really benefit from receiving chemotherapy. Likewise, in this intermediate recurrence score group, again, the yellow and black lines here are quite similar. The black line is actually the chemotherapy before tamoxifen. You can see those patients went through this extra chemotherapy but didn't have any clinical benefit from it. And it's these patients that are specifically in this high recurrence score result range. So they did the Oncotype DX. They had a high probability of recurrence based on that 21 gene signature. These are the patients where the outcomes for the chemotherapy before tamoxifen are significantly better than with tamoxifen alone. So I think one from this, you can start to get a sense of the value of this and the important goal of avoiding overtreatment. So if, one approach is just give all patients chemotherapy plus tamoxifen, the majority of those patients, you can see from this data, are not going to benefit from the additional chemotherapy, the cost, the side effects, all the negativity associated with having to endure chemotherapy for no benefit can be avoided by potentially using this genetic signature to identify the patients that are most likely to benefit from that treatment. There still are questions and uncertainties around this test, and I would say that the data from the clinical trials and the outcomes from it's really as good as the data. And for all these clinical trials, one has to consider what's the population that's included in the clinical trial and how does that relate to the individual that's trying to make a decision about whether to implement this test. And so I think what I've read about this particular test, and I'm not a clinician again, but what I've read is that some underrepresented groups that are not highly reflected in the trial populations, it remains uncertain how well the test reflects the likelihood of recurrence. And so young patients, patients with BRCA mutation, Black patients, elderly patients, those are the groups that, as I understand it, are less well represented in the trial data. And for those groups, there might still be questions about whether this is going to be a test that's really predictive of recurrence rate. But for certain individuals, clearly there's a benefit that might encourage certain individuals to get chemotherapy because they're likely to benefit, or to avoid chemotherapy because they're unlikely to benefit. Okay, I'm going to give another example related to another kind of similar test. This is called the Breast Cancer Index. And this, again, is for estrogen receptor positive patients. And it's really focused on the question of whether it's useful to extend anti-estrogen therapy beyond 5 years for early stage hormone receptor positive breast cancers. So I think it's likely that many of you are aware that recurrence after anti-estrogen therapy is not uncommon, and it often happens at later stages. It can be 10 plus years after treatment, and it would be useful to understand which patients are at highest risk for that relapse or recurrence event to occur. And if those patients can be identified in advance, one could extend the duration of the anti-hormone therapy and reduce the risk of recurrence. There are challenges with extending endocrine therapies. One can't just do this to everybody or shouldn't do this to everybody, because those patients that aren't going to benefit from it still are going to experience the side effects, which can include they're listed here: cardiotoxicity, bone pain, fractures, menopausal symptoms, etc. So there are negatives, downsides to extended adjuvant endocrine therapy. So the goal of this test is really identify those patients that are going to benefit from that therapy and avoid a late recurrence or relapse, and then spare individuals that are not going to benefit from it, from those negative side effects of the extended therapy. And so this particular test is based on a couple of parameters. It really makes use of some observations that high levels of the gene HOXB13 and low levels of another gene, IL-17BR, are associated with recurrence. So one can measure this ratio of HOXB13 to IL-17BR, and this is both prognostic, it predicts the risk of breast cancer recurrence, and it's predictive of tamoxifen benefit. This ratio is actually particularly effective for identifying late recurrences. And then this ratio is measured in this assay together with an additional 5-gene panel. Those genes are listed below here. You can see so the HI ratio that I just mentioned, plus these 5 genes, BUB1B, SENPA, NEC2, RAC-GAP1, RM2, that's this molecular grade index, which kind of gives an indication of the molecular grade, how aggressive a cancer is going to be. That particular signature is not as effective for its determining late recurrence. So it's coupled with this other HI index to have an overall score of the breast cancer index. And then there are these additional reference genes that are mentioned. And the technical approach is similar to what I just described. So it's this use of RNA that's isolated from these fixed paraffin-embedded samples. And then that RNA is converted to DNA, and then that DNA is used for a PCR analysis to measure the expression levels of all those genes in the panel. And this test across multiple clinical trials, which are listed here, does seem to highlight individuals. It does have predictive value for identifying individuals that would benefit from extended anti-estrogen, anti-hormone therapy. And so, as one example, this MA.17 trial with 249 patients. So patients here received tamoxifen treatment, and then the clinical trial had a comparison of 5 additional years of aromatase inhibitor versus placebo. And the outcome was that individuals that had a breast cancer index value that was high, predictive of a strong likelihood of recurrence, showed a 65% relative risk reduction when they received this extended aromatase inhibitor treatment relative to the controls. If the breast cancer index said, no, this is not a candidate for this, they're not likely to recur, they observed no significant benefit associated with the extended aromatase inhibitor treatment. And you can see similar outcomes with different trial designs where instead of aromatase inhibitor, it was additional tamoxifen treatment, et cetera. But the net effect here suggests that this breast cancer index can identify individuals that will benefit through risk reduction by extending hormone therapy for additional time. And for those individuals, the downsides of the additional hormone therapy are likely worthwhile, given that the risk of recurrence is going to be reduced. Okay, so both of those assays that I just described used tissue that was collected directly from the tumor for analysis. So in both cases, the tumor section was recovered and molecular analyses were done. In the final portion of the talk, I'm going to talk about a series of tests that can be done with other sources of material. And this table is one kind of introduction to this concept that compares the utilization of what we call circulating tumor DNA or ctDNA versus tumor tissue testing. So tumor tissue testing, a tissue-based assay, the features of it are listed on the right. So here, this is invasive, right? So the tissue through a surgical or other invasive procedure is removed from the patient. It's more challenging to obtain. There can be risks of biopsy procedure. The serial testing of the same patient repeatedly is more difficult because each time you're going in to take directly tissue from that individual. The biology, one can visualize the histology of the tumor and correlate that with whatever readout. That can be a useful thing. However, the region that's being assayed here is whatever piece of tissue that's collected. So if a large tumor is surgically removed and one area is sampled for histology, and that's what's used to generate the molecules in these assays, they're really only sampling that one small piece of tumor. The alternative here are these circulating tumor DNA assays, and we'll talk about this more, but this refers to DNA molecules that are present in the bloodstream or from other fluids or other modes of collection. I'll give you some examples of that. Blood would be the most common. These circulating DNA molecules, I'll talk a little bit about their origins, but they're derived from the tumor cells and are present in the bloodstream or other fluids. So you can imagine in the blood, rather than taking a biopsy of a tumor tissue itself, now we're just drawing blood. So this is easy to collect. There's, some risk of blood collection, but typically minor compared to biopsying a tissue. And it's relatively easy for a patient to come in, give a blood sample, and to do serial testing over time with a less invasive procedure than a tissue biopsy. The biology here, the readout, the molecular readout, it doesn't directly correlate with histology or cellular phenotype because we're looking in molecules in the blood as a readout for the tissue, but we're not looking directly at molecules from the tissue. So it's harder to correlate directly with histology, but at the same time, it's more likely to represent the whole tumor because we're not looking at a specific portion of the tumor. We're looking at tumor DNAs that are found in circulation. Okay, so I'll use this as an introduction, but I'll get into this in a little bit more detail. And the general area here we can refer to as liquid biopsy. So this is the idea that instead of directly biopsying tissue from a tumor, we're taking a liquid biopsy in the form of blood or other fluid, and we can find many tumor-associated molecules in that sample and use those for diagnostic purposes. So here's a example of a blood vessel from a patient. We can see red blood cells, but we can see DNA. We can see actual circulating tumor cells in the bloodstream, and we can see other molecules and other entities released from the tumor in the bloodstream. We collect those in the blood sample, and then we can look at all kinds of genomic alterations in these samples as a readout for what's going on in the tumor. And I thought I'd give one example. This is not a breast cancer example. It relates to work happening in my own laboratory, and it is relevant to the topic. So I thought I'd just briefly introduce this. My lab has an interest in germ cell tumors, which are most common in men. So we study commonly testicular germ cell tumors. One common type of testicular germ cell tumor. This also has relevance to ovarian germ cell tumors. They're rare ovarian tumors, relatively infrequently compared to the ovarian cancers of epithelial origins, but still a clinically important area of ovarian tumor biology, less common than the testicular germ cell tumors that are the focus here. One type of testicular germ cell tumor is called embryonal carcinoma. And one challenge in the field over the years has been the poor performance of classic blood biomarkers. And you'll see some of them, you might even recognize some of the names like AFP, beta HCG, LDH. These are biomarkers that are used clinically for patients with testicular germ cell tumors. You can see for embryonal carcinoma, their sensitivity is really poor. So, 20 to 40% of patients with one of these cancers will be positive for one of these biomarkers. So lots of potential for false negatives because less than half of the patients are going to show a positive result. And then these are not very selective also, 10 to high levels of detection in patients that don't have testicular germ cell tumors. So the two features we look for in a biomarker, sensitivity and selectivity, these are not very good. What's emerged in recent years is that a special class of RNA molecules called microRNAs dramatically outperform these classic serum biomarkers. So these are small RNA molecules that can be detected in the serum of patients, and over 90% of patients with this kind of malignancy will be positive for this biomarker, and the detection in non-testicular germ cell tumors is very low. So this is really kind of revolutionizing how we can perform diagnosis on testicular germ cell tumors. It holds promise for doing that. I think it's in the transition now. There are clinically approved tests to measure microRNAs like miR-371, and it's kind of getting ramped up through clinical trials and continuous assessment to see if this can be a tool that can be used to help diagnose patients. I think the results, and as you can see in this table, look very promising. I think for sake of time, I'm going to skip the biology of why this is such a good biomarker specifically for these cancers. This is a relatively unusual thing where a single microRNA is such a remarkably good biomarker. It relates to the biology of these tumors. I won't go into that. But I also wanted to make a case, and this really illustrates it well, excuse me, for the importance of basic research. And I note it in this example because the biomarker here is a microRNA. And these two gentlemen, Victor Ambros and Gary Ruvkin, received the Nobel Prize for their studies of microRNAs just last year in 2024. And the discovery of microRNAs came from their work in a soil worm called C. elegans. And they were studying mutant worms that developed abnormally. And they were trying to understand the biological molecular basis for these defects in worm development. And through that work, they discovered this whole regulatory mechanism based on microRNAs. So before their work, microRNAs were not known. They didn't be known to exist. So these guys discovered microRNAs and received the Nobel Prize for that work. They did this again in this really obscure worm model of embryonic and worm development. They weren't thinking about a cancer biomarker, but those same molecules now are this really promising marker for a human cancer. And I think this is really great illustration of why basic research is so important. We obviously need to do applied and translational research, but we need to understand fundamental biological mechanisms, which ultimately will fuel translational and clinical work. And this is repeated over and over again in the literature. The studies of checkpoint inhibitors that we use for immunotherapy came out of basic studies. Things like CRISPR that you probably just heard recently, their clinical utilization of CRISPR to correct a metabolic defect in the liver of a human baby, right? This technology came out of the study of bacteria. So I just want to make a case that basic research really drives all of our subsequent clinical research, like all the work I'm talking about today. Okay, so back to breast cancer and gynecological cancers and the role of these liquid biopsies. So here's another illustration of just the general concept. So we can take many different kinds of biological specimens from patients in a non-invasive way. So blood is probably the most common, but others like saliva and urine. And those samples will contain a variety of molecules that have diagnostic value. In some cases, it's DNA. Some cases, it's protein, microRNAs, like we just heard about. All of those things can be measured in these samples, and that can have a lot of utility for early detection of cancer, detection of tumor recurrence. So we can detect these molecules in a much more sensitive way than traditional approaches where we maybe were imaging to detect recurrence of a tumor. We can also detect very quickly how well a patient is responding to a therapy. We can also make determinations of prognosis and outcome based on the particular signatures of particular molecules that we can find in these liquid biopsy specimens. So again, just a long list here of potential sources for these materials, which can vary depending on the tumor type of interest, and then many different molecular approaches and analyses that can be done to look at features like mutations, copy number changes, changes to epigenetic features, not only the DNA sequence, but methylation of DNA, other features of the DNA that, again, can correlate with biological phenotypes and can predict outcomes or response to therapy. Okay, so the most common of these that we're going to talk about is circulating tumor DNA. You might see on some of my slides or read about this, also called cell-free DNA. So cell-free DNA would be any kind of DNA if we just focus on the blood. If there's any free DNA in the blood, not within a cell, that's cell-free DNA. That is a broad category. So normal cells can release DNA also. Circulating tumor DNA would be cell-free DNA specifically derived from a tumor cell. This can happen when cancer cells die and they can release their DNA. They can also shed or secrete DNA through other means that I'm not going to go into in detail. But all of these things, the net effect is they result in DNA present in the bloodstream or in other fluids. Then it's possible to analyze that DNA through different approaches. This can be the PCR kind of amplification assays that I mentioned that are gene specific, or they can be genome sequencing assays as well. And these approaches can detect mutations at specific DNA sites, and they can detect other modifications like DNA methylation. In addition to cell-free DNA or circulating tumor DNA, which will be my main focus, there are other secreted and other molecules and even cells present in the bloodstream. Some of the assays focus on circulating tumor cells themselves. So this is an example here. We have primary tumor site and we have tumor cells in the process of metastasis go into the bloodstream and travel to different sites. But through this process happening, there are cancer cells present in the bloodstream. Those can be separated, detected, and that can be another diagnostic, the number and features of cancer cells present in the bloodstream. And then finally, there's another category of small particles secreted by cancer cells that can also be found in the bloodstream. And I'm not going to go into this in detail, but this is a normal biological process. Normal cells shed these molecules called exosomes, and cancer cells do as well. They're small lipid-bound particles. They're full of RNA, DNA, protein, and other molecules. These exosomes can be collected and then used for diagnostic purposes. And one thing I mentioned before is that these circulating tumor DNA or cell-free DNA assays are not restricted to a particular piece of tissue that's been collected, but really reflect the entire totality of tumor cells within an individual, proportional to the number of those cells, but they're all releasing DNA. And so this can be a useful approach for detecting events at different sites within an individual, and also even heterogeneity within a single tumor. And that's just illustrated here. So this individual, for instance, has this large tumor in the lung in blue, and then an additional lesion in red there. And then over time, with treatment, the blue shrinks, this new green tumor arises. With a different treatment, these shrink, both shrink, and the red one disappears entirely. So those collective changes can be monitored in the blood because each of these sites is going to, in theory, be secreting, releasing DNA that can be monitored in the blood. These different sites might be associated with different molecular features, different DNA mutations that allow them to be distinguished in the ctDNA. And so we can get a bigger, more complete picture of what's happening in a patient than looking at DNA from, say, a single biopsy. And then here are examples, again, of genetic testing for later stage cancers that often can make use of these circulating tumor DNA kinds of assays. And I'll just go into a couple of specific examples, one of which relates to monitoring mutation in the estrogen receptor, or ESR1. Excuse me. So estrogen receptor is prone to mutation that causes resistance to endocrine therapies. So the estrogen receptor normal function is shown here in the left, the wild type. The hormone in blue here binds this protein that influences its ability to bind DNA and act as a transcription factor. Molecules that anti-estrogen therapies can either prevent the generation of the estrogen or act as a competitor that prevent it from binding, or bind in the same site and target this receptor for degradation. Resistance mutations can arise. These initially actually were missed because many of the genome analyses were done with treatment naive samples, but then it later emerged there are these really critical mutations in the estrogen receptor that essentially allow the estrogen receptor to be active even in the absence of estrogen. If, for instance, one is on an aromatase inhibitor that's blocking the production of estrogen, these mutations will essentially allow the estrogen receptor to continue to function even in the presence of the aromatase inhibitor. So these are mutations that it's useful to know are present, and is useful to know as soon as they arise, because they're going to influence response to therapy. So aromatase inhibitors shown here are going to be ineffective in an individual with one of these mutations, and certain other anti-estrogen therapies also are going to have potentially moderate activity, and other approaches are going to be more effective. So through these kind of assays that monitor the presence of these mutations in circulating tumor DNA, one can detect these events early on. So here's just an example of a kind of schematic of a circulating tumor DNA measurement. With surgery, the amount of circulating tumor DNA is going to drop dramatically. If this patient received aromatase inhibitors, the values stay low, but then this estrogen receptor ESR1 resistance mutation is detected. And upon detection, this could drive a change in clinical approach with recognition that this mutant version of the estrogen receptor is no longer going to respond to aromatase inhibitor, and it's better to choose, for instance, an estrogen receptor degrader as a different approach. And kind of similar approaches can be used, and I won't, for sake of time, go into the details here. So one assay that's focused on circulating tumor DNA that people hear a lot about these days is the Signatera assay. So this is an individualized circulating tumor DNA assay. Basically, it uses genomic sequence both from a tissue sample and a blood sample to identify mutations present in that particular DNA, the DNA from that particular patient. That information is used to make a custom panel that's personalized for that individual. And then those mutations that were present in these tumor samples then can be monitored subsequently, for instance, following surgery or treatment to monitor, for instance, for recurrence. So if we see this kind of from the Natera website, how this assay can be used. So initially, mutations arise during the evolution of the tumor. The prognosis and staging is done. At that point, the tumor gets sequenced. The blood samples get sequenced to identify the mutations that are indicative of this particular tumor. After surgery, the levels of the circulating tumor DNA drop, but then continued monitoring with that same panel is done to identify recurrence. And so here we see that relapse has happened, and this might allow for early detection of relapse. It might allow for now a new treatment that would take into consideration the resistance mechanism and monitor whether that is working. So here's one clinical study with this approach. This is essentially the Signatera approach. In this particular case, it's 156 patients with primary breast cancer, followed up to 12 years with semi-annual blood sampling. And here's the distribution of different breast cancer types. So each patient got its own, each patient received, tumor sample was used to design an individualized panel with 16 mutations. And then that approach was then those 16 mutations were monitored in that individual across semi-annual blood sampling. And this approach of circulating tumor cell DNA detection allowed detection of relapse in 30 of the 34 patients who relapsed before clinical or radiological relapse. So anywhere from 0 to 38 months ahead of the traditional approach to detect relapse, median of 10.5 months, this blood assay was able to detect the presence of the cancer coming back. And here's some more clinical data, just again, showing that the presence of the circulating tumor DNA is indicative of relapse and patient outcome. So it is an early indicator of the disease coming back. There are, again, it's not a perfect assay. In this particular cohort, there were some patients that developed recurrent disease despite being negative for this ctDNA. So this would be false negatives. And then there were also some false positives that had positive ctDNA, but they weren't diagnosed with recurrence over prolonged follow-up. Okay, and I think one last point, and then I'll wrap up. I know we're running late, relates to this last point on immunotherapy. Since this is an exciting area in cancer research and cancer care, I thought I'd say, just note that there are these assays to measure biomarkers for immunotherapy response. I'll note one that is used for patients. It's tumor mutation burden, so the number of mutations in a tumor. And this generally correlates with response to therapy. So number of somatic mutations along the x-axis, response rate to checkpoint inhibitor treatment on the y-axis. Generally, the more mutations in a patient tumor, the better the response to immunotherapy and checkpoint inhibition. And that's because these approaches of immunotherapy really rely on the immune system being able to recognize mutated proteins within the cancer cell in order to kill the cancer cell. So the more mutated proteins, they're often referred to as neoantigens, new antigens, that the immune system can detect in a cancer, the more likely that the immunotherapy is going to work. There are many other parameters besides tumor mutation burden that influence how well the immune system is going to be able to work. And in breast cancer, this has been a big challenge to make immunotherapies work better. And so I think this will be an exciting area for future research. And I think with that, I'll just end and be happy to answer questions.
That dredged up some memories of my biological sciences initial major before I switched. But those pathways, they were still in there, buried deep, but in there.
Good.
So I'm starting to remember some of the terms that you used. And we did get some questions in the chat, and it looks like I will try to keep on top of the questions as people are putting them in as best I can. But there were a couple questions submitted in advance about liquid biopsies or blood biopsies. So they've been making headlines, and you did sort of share the lay version of their impact. But the question was, is overtreating a concern? And what about the policy impact of quickly having Medicare cover them? It sounds like they were sort of rushed through to be covered by Medicare, but then as a result, there's a concern of them being overused. And there were some other questions about, they're sort of counterindicated for breast cancer in particular, and maybe other cancers as well. So curious on your thoughts.
Yeah, so I think I would again follow clinical recommendations, and national organizations like ASCO, I think, are the best source of information about what's clinically appropriate, and I think physicians will tend to follow that recommendation. I think the promise of these, like I discussed, is that they ultimately can help identify and distinguish patients that would benefit from a therapy versus ones that will not. And I think that ultimately will enable a reduction in overtreatment of patients, where a prolonged treatment or addition of additional therapies through these assays, one can distinguish who's going to benefit and who is unlikely to benefit. And that will, I think, allow for more precise and appropriate use of treatment. I think, right, there's a balance between having the appropriate amount of information to make these clinical decisions and then overdoing it, right, in a way that's not productive. And I'm not sure we have enough information yet from clinical trials to know when that's the case.
I'm muted. So more information needed. We need some more time, especially for these relatively new tests. Okay. Yeah, so far I think I've got 11 questions, and we'll get through as many as possible before 8:30. We're going to do it. All right. Let's see. There was a question about genetic mutations, such as the ATM gene or other mutations that are designated as a VUS or variant of unspecified significance, and wondering whether those sorts of genetic mutations are anticipated to further mutate and possibly cause a future recurrence. And I guess that mutation in particular, and some others, prophylactic surgeries are recommended with those genetic mutations. So it changes the decisions that people make about surgical options.
Yeah, I can comment on this this general area. So I mentioned how in our DNA sequence, we've inherited these variations in different genes. Some of the variations are clearly known to be what we call pathogenic or disease associated. So a certain mutation can be well characterized to, so there, for instance, in the gene that we just heard about, ATM, there are well-known pathogenic mutations that disrupt or impede the function of that gene and that are associated with cancer risk, and those are well characterized. Then there are other DNA sequence changes in the same gene where the functional consequence of that is unknown or not clear. So those would be called variants of unknown significance, this VUS moniker. So those are ones that there's a change in that gene. We don't know. It could be a change that doesn't have any effect on the gene, or it could be one that's harmful that would be associated with cancer risk. So there are some computational predictions that one can make to determine, is this variant associated with risk? And then there are experimental approaches, like we can model some of these in the laboratory and determine, does that negatively impact the function of the gene? Or we can look at an array of human data and see if there's an association. Do people that have that particular variation, are they associated with risk? So I'd say between those different things, modeling and predicting, I'd say many times we can make kind of educated guesses of whether something is going to be associated with increased risk, but there are some where at this point we still don't know. And then I think in those cases, one needs to be cautious about how to use that information, because it could be that it is a change that happened that isn't going to influence cancer risk. It's called a VUS because it is unknown.
Yeah. And yeah, bearing in mind something you mentioned earlier about the environmental factors and the behavioral factors. So it's not solely genetic information in isolation. It always exists in a context and in an environment. And so there's so many variables. It's hard to isolate. I mean, it's easier in a lab, I'm sure.
No, you're absolutely right. It's complex and it's multifactorial, and that's why some of those like risk calculators try to bring in all, like, it's many genetic features, it's many environmental behavioral features, and people are trying hard to, how can we create algorithms that kind of put it all together to, and I don't know where those are at in terms of variants of unknown significance. Maybe there's a place in those calculators for that kind of information too. I don't actually know.
Potential helpful use of AI in the future, maybe.
Absolutely,
We'll
yes.
see. If it can help us, that would be great. All right, let's see. Um, there were a few questions about timing of testing.
Hmm.
Someone asked, like the BCI, the breast cancer index, should it be done at the start of endocrine therapy or at 5 years after treatment? There was another sort of more broad question about how long after diagnosis should any of these tests be done? 5 years, less, longer, ongoing?
Yeah, good question. And I don't know the details for many of those. I think the breast cancer index in particular, my understanding is that's done basically prior to onset or at the time of therapy. I think the original features of the tumor, I believe, are predictive in that case. It would be best to, again, consult your physician about that. I think for the um, other tests that based on circulating tumor DNA or cell-free DNA, those can be done on an ongoing basis, and those are more for the purpose of monitoring for recurrence and response to therapy.
Okay, that's helpful to know. And it sounds like often talking to your oncologist or your team on their recommendations is, yeah, is advised.
Absolutely.
Um, okay. Another question about, are there any recent advances in testing for triple negative breast cancer, which is a more rare kind of breast cancer, and even more specifically for someone who is BRCA or BRCA positive?
Yeah. So, I mean, I think the assays that I've described um, are, I mean, not, there are certain ones I mentioned that are specific to hormone receptor positive cancers, but like the Signatera assay and others are appropriate for all types of breast cancer, including triple negative. I think there are well-established assays from Myriad and others for the germline BRCA variants, and where there's a family history or other considerations that might suggest this is a factor. I think those are recommended in those cases.
Um, Someone
And
asked,
also, sorry, and
oh,
those can also be ones that direct patients to particular therapies also. So there are, we haven't gotten into that too much. Occasionally it popped up today, but for BRCA mutation, cancers of BRCA mutations, there are PARP inhibitors and other strategies that those cancers are particularly susceptible to. And so knowledge of status of those kind of mutations can be very impactful for directing decisions about therapy.
there was a question about um, whether saliva testing is more accurate than blood testing, and whether that is new testing since 2017, which I think brings up a question also about, just how many mutations we are aware of with each year that passes. Like for somebody who got genetic testing and knows about, maybe they tested negative however many years ago, like in 2017, is it recommended for them to retest for their genetics today in 2025 and then figure out these different assays that they could do depending on the results?
Yeah, I mean, the technology is certainly advancing at a really remarkable rate. So the number of genetic features that can be tested in these assays and not only mutation, but other parameters. We didn't talk about it much today, but there in cancer, right, we have changes in the DNA sequence, but we also have what I touched on very briefly as these what are called epigenetic changes. These are also changes that influence the expression of genes that are heritable. So they're passed on, but they don't change the sequence of the DNA. They change the way DNA is packaged or modified. And there are assays now that can also detect those kind of changes like DNA methylation or related changes in circulating samples. They're very sensitive, so they can use very small amounts of material and give really astonishing insights into what's happening at the molecular level. So I think these things are evolving, and I think each generation of these tests provides new insights for sure. I think for the saliva versus blood, I don't actually know I imagine there are some direct head-to-head comparisons out there in the literature. I'm not familiar, and each of these I think you have to think about uh, like the placement of the tumor and how likely it is to release molecules into a given fluid. So is a tumor more likely to release cells or DNA into the blood versus the saliva based on where it's positioned? And then also the material itself can influence the stability of molecules. So the pH and other features of blood versus saliva is going to influence how stable things are, and that's going to influence these readouts as well.
This is so fascinating, I just have to say. Okay. When will these circulating DNA assessments begin on a regular basis? What are the best questions to ask your physician about your individual therapies? So, like, do you have any recommendations for folks who are going to be seeing their providers and, they just learned everything in this webinar. It's a lot of information and they have questions, how best to ask about them in a way that
Yeah,
maybe, you know, there's that delicate balance between strong self-advocacy and, providers are people too. They also have egos. So without being off-putting, do you have any
Yeah, I mean, I
advice?
think, going to these kind of sessions, reading about these things, so that you go into those appointments well informed is good. And so doing some additional homework on this, maybe related to if one knows what the features of their, what the cancer type is, what stage it is, that can influence which of these tests is appropriate or not appropriate. I think I've always been familiar with the recommendation to, bring a family member or friend along that can help with the conversation, help with note taking and remembering what was said in these conversations. I think that's generally good advice as well. I think it's appropriate to ask, what kind of testing is available for my tumor, what mutational analyses, and I think in some cases, advocating for oneself and pushing for these things can be a good thing. I think more information often is helpful, but it's important to do that in a kind of collaborative way with the clinician, so that you're kind of working together as a team.
I wish we could just take you with us. That'd be so helpful. Okay, two more questions, I think. Um, for folks with metastatic or stage 4 cancers, scans can indicate no disease progression, and yet tumor markers can remain abnormally high. Is this to be expected, or should this be cause for concern and, requesting additional tests?
Yeah, so that's certainly the case. I think these molecular analyses, typically, for the most part, are going to be more sensitive than some of those imaging modalities. It does suggest, it can suggest, and depending on what markers and how many markers, it can suggest the presence of disease. Maybe at a lower level than is detectable by imaging. I think it would have to be an evidence-based approach to know what's the level of marker detection in the absence of a imaging result that warrants additional intervention. So I think it's probably going to be very case specific in terms of which markers and what the scenario is and what the options are also, because I think yeah, there would be considerations about what treatments are even possible at that stage and appropriate, that would need to be considered.
Thanks for that. And then the last question. So how does this awareness of recurrence influence overall survival? I think, something I learned fairly recently um, at the National Breast Cancer Coalition Leadership Summit is that sometimes the measurement of progression-free survival versus overall survival isn't the same thing. I don't know if folks in this audience are familiar with the two different terms, if you've read any studies, but um, overall survival seems like the most important measure of period. Um, so, maybe sort of like a really brief explanation of, yeah, whether recurrence is necessarily a good indicator of that. And so, the testing to try and um, detect things sooner, is there a correlation and causation with, yeah, how overall survival is measured?
Yeah, and I think there are some indications that that's the case. And I think if you think about particularly the relapse associated with resistance mechanisms where there are alternative therapies in place, the prediction would be that, right, if one detects relapse sooner through a molecular technique than later with some other approach, that if that can drive the change in a therapy, that would be predicted to lead to improved long-term survival and overall survival. Again, it depends on a number of parameters, including whether there are therapeutics available that can overcome a resistance mechanism, for instance. In some cases there are, in some cases there are not, based on where we're at in terms of developing therapy. So I think in some cases, I think it is a useful a useful parameter to enhance overall survival. But again, it depends on the availability of therapies.
Okay. Well, we always seem to run out of time. And, I feel like we're just really getting going and digging into this, but it has been illuminating. Thank you for sharing an hour and a half with all of us. If anybody wants to rewatch this, which I might, because it was a lot of material, very dense, and I wish we could just sit in on your classes, Dr. Weiss. But let us know in the future if you want to learn more about these sort of scientific advances in that little survey that I mentioned that's going to pop up as soon as you leave us. And hopefully we can all meet again on Zoom next month. And I look forward to seeing you again, hopefully at the coalition at some point. Bob, thank you so much.
Sounds great. Thanks for having me. Thanks for all the good questions and discussion.
Yeah. And take care, everyone. Hopefully see you all soon at the coalition as well. Bye-bye.